Thank you, Kristen, for the introduction. So the image that I want to talk about, the story I want to talk about today, is one about engineering the plant-to-microbe communication. In nature, plants and microbes naturally communicate by sending each other lots of chemicals. It's kind of like sending text messages, but with chemicals instead. And what we're trying to do here is to create a new communication signal between plant and bacteria that none of them can really sense otherwise. And we want to do that so that the microbes can help keep the plant healthy, but also detect things like landmines or heavy metals to help degrade them and restore contaminated landscapes. So how do we do this? We want to have a modular structure so that our microbes, our engineered microbes, can do different things and perform different tasks. So we want them to be able to sense different types of environmental stimuli, such as plastic. It could be nutrients in the soil, heavy metals, or pathogenic fungi, for example. And we use this by using a sensing computing output framework, which is kind of very widely used in synthetic biology. And so the bacteria will be sensing the stimuli, and then in response of it, will produce an output. And in this case, the output will be a bacteria-to-plant communication, so a molecule that will diffuse out of the bacteria and into an engineered plant cell. The reason we're using an engineered plant is so that no other plant will be able to detect the signal, only the plant that we're interested in. And so upon receiving the signal, the plant will produce an output. This output can be quite varied. It could change the plant phenotype, it could improve plant growth, boost the plant immunity, or degrade heavy metals, for example, that are in the soil. But for now, we're just focusing on changing the plant phenotype. We're looking at producing GFP, which comes from jellyfish and fluoresces and makes our plant green. So I'm going to focus on one specific example because we've been working with different plants and different types of bacteria. But this is an example where we're using a bacteria called Pseudomonas butida, which is a common soil bacterium, to detect arsenic, which is a heavy metal widely found in the environment that's toxic and causes quite concerns to human health and wildlife. So we've engineered our Pseudomonas butida to produce our bacteria-to-plant communication signal, here PCHSL, which we're producing through a 3-gene pathway. This molecule will diffuse out of P. butida and then be imported through just natural diffusion in Arabidopsis thaliana, which is a model organism used in plant biology. We know a lot about it. You can see it, a small version of it in well grown in hydroponic culture. And as you can see in the microscopy images, the bright field images shows the white structure of the roots. And when the bacteria and the plants are grown together with our arsenic, you can see that we're not detecting any green fluorescence signal in the root. But when the bacteria and the plant is grown together with arsenic, you can see a diffuse GFP signal along the root network. And you might say, well, this is great, like GFP is turning out in the root, but we don't actually normally see the root. When you look at a plant, you only see the arrow part of it. You only see the leaf and the flowers. What we're trying to do now is expand this work to be able to look at things in the leaves. So if you look on the left, the image is more in the violet blue tones, while on the right you have more orangey tones, which means that there's more GFP intensity that we're detecting in the leaf of those plants. So this still needs some optimization, and we're looking at using reporters that don't necessitate special equipment. So no fluorescent reporter, but pigments, which you would be able to just look at by looking at your plants. So with that, I wanted to acknowledge my PI and Dr. Tyler Toth, who worked on this project, and the whole Voight Lab for their incredible support. The imaging centers and the funders